presentation. All right, everyone, welcome to Farms Weekly Wednesday webinars. This webinar series is sponsored in part by Farms with a grant from the Otto Bremer Foundation. And if you go to our website at www.farms.org, you can see the rest of the offerings for this year. We will go through May. Our webinars run from 12 to 1 so that people can view them while they're at work and get back to work by 1 o'clock. So we start promptly at 12. Everyone will be placed on mute or can place themselves on mute during the presentation. If you have questions that are pressing, please use the chat box, and when Frank takes a breath, I will step in and ask a question. If not, if we could wait till the end of the presentation to ask questions, that would be super cool also. We do record these sessions when it works. I apologize again for last week's terrible fiasco with that video that we were going to show. I wanted to report to everyone who was on that I'm trying to get a hold of that video because NDSU had asked Irene to remove that from their site because it was quote unquote unresearched and in an unapproved facility. So they didn't want anybody dying by using that video to cure their own ham and bacon. But we're going to see about getting that up on our YouTube website, and I'll send out a link to that. If you get an email after this session, which I think you do, that says, please fill out this evaluation, it will really help us a lot with future funding and future programming. Frank, I've known Frank since he worked with Sarah in some region, the north. I don't remember what region we are, but I'm going to allow him to tell you more about what he's doing now. He currently works part-time for NPSAS and also as his own farmer breeder type person. So I'm going to change presenters. We're going to make sure we can see Frank's screen, and then we'll let him take over. And you should be getting a little box right about now, Frank.
Okay. Is that working?
Yes. I can't pronounce the word Gametophytic Incompatibility, but I can see your screen. So you go ahead and take over. I will mute myself, and we will begin today's presentation. Thank you, Frank, and welcome.
Well, thanks so much, Sue. Here, I'm going to try to get this out of the way, too. And I think I just do that. There. So, yes, as Sue said, I'm Frank Kutka. I did work for the North Central region of the Sustainable Agriculture Research and Education Program as a extension specialist with North Dakota State University for quite some time. I currently am a co-coordinator with the Northern Plains Sustainable Ag Society's Farm Breeding Club, and we have a lot of interesting projects. Most people who know me as a corn guy, and I have been doing corn breeding and some corn genetics work at home for really just a little over 20 years now. Got interested enough as a hobby to actually go get a degree in breeding, which opened up a lot of interesting possibilities. And today I'm going to talk about what it is that gametophytic incompatibility is, besides a mouthful of syllables. So, as you may or may not be aware, there are people out there who keep their own seed corn and have for a very long time. Both Native Americans and immigrant farmers as well have a lot of seeds that they save. It's not nearly as huge as the ethanol corn production system or anything, but there are people who've got these varieties out there. Some do their own breeding at home and are breeding new varieties of corn. This is Dave Christensen's Painted Mountain flower corn from Big Timber, Montana. And as you can see, it's remarkably prettier than most commercial corn that you would see. Kind of interesting stuff. Some corn out there is nutritious and delicious. So regardless of what you've heard about commodity corn, that is not the whole corn story. Commodity corn these days really isn't running very high in protein, whereas corn traditionally went anywhere from about where it is now up to about twice as much protein as it has now. So In the old days, it was not unusual to come across varieties that had 10 to 14% protein, high oil, and high minerals, various traits that you don't normally think of in corn these days, but some does have this, and people are quite pleased to grow it. And if you're not aware, growing corn commercially under organic certification is also pretty valuable. It's another thing people do with corn that's not quite the mainstream. These are USDA data from the last couple of years, and you can see in the last few years, organic corn prices have always been, at least $10 a bushel, and have been as high as just over 14, which is pretty remarkable for just number 2 yellow dent hybrid corn. But in all of these cases, anywhere where people save seeds or grow corn for special traits, if they're growing corn they want to maintain, there is this issue of cross-pollinating and getting seeds that have these transgenes from transgenic corn. There is a misnomer out there. A lot of people call this corn GMO, or genetically modified organism corn. But genetic modification is not really a very specific word. It doesn't mean much, whereas transgenic means that we brought these genes in from another species, and that is precisely what we're talking about. These genetically engineered gene or DNA sequences that are inserted into the plants and are patented and then sold by the very large seed companies. Well, corn is a wind-pollinated plant, and pollen does blow from these fields of transgenic corn, which can number in the thousands of acres or wherever you live, and it can blow into your organic or traditional or home breeding project. And what can you do about this? Well, of course we can try to plant corn farther away from other corn. We can plant corn that flowers at different times. We can use border rows, but I guess we should take a look at the corn plant itself, and then I'll explain more about gametophytic incompatibility and how this fits in. The corn plant is a grass plant, so it has a main stalk that has nodes and internodes, and at each of the nodes a leaf protrudes. Most modern corn is single stalked, but it doesn't always have to be, and certainly wasn't always in the past. It has imperfect flowers. The flowers usually don't have male and female together. They're usually monoecious, and they also don't have petals. They don't have sepals. They don't have a lot of those things that we normally think of when we think of flowers. However, we do have these female flowers we call the ear shoot, and we have male flowers that we call the tassels. Now, in each case, these are multiple flowers all massed together in a large aggregation, if you will. The ear is a large number of individual female flowers, each with their very own silk, which is the stigma. It's the part that catches the pollen. And then the tassels are large masses of individual male flowers, each covered with a glume, and from which we see a number of anthers will protrude and release pollen. The tassels usually come up first, and the anthers usually open up first, but within a few days the silks will start to show, and pollination will go forward. The pollen blows on the wind, and when it reaches a corn plant that has the silks out, the silks actually have little tiny branches, and they're very, very wet, multicellular but very skinny structures. And a pollen grain will land on these, find the right signals for germination, and it will germinate. And I'm using the term germinate because it's really kind of what it is. A pollen grain is sort of a spore, if you will, and it grows its very own plant, the pollen tube, which is a gametophyte, which means it's a separate life stage in which we've got gametes, or sexual reproduction is going to take place. So the pollen grain lands and it's going to grow this little plant, this long tube down the silk. And the remarkable thing is, as skinny as these silks are, and as long as they are, that pollen tube from that tiny grain of pollen has to grow the whole length of the silk. So it needs to be supported to do that. It needs to get fed. It needs direction and assistance to figure out which way it's going, so it can get all the way down to the individual flower where the egg sac is, and then the DNA from the pollen tube and DNA from the nuclei in the egg sac, these can join together and form a new corn plant. So a seed will be born and will develop right there on the ear. Now, you could try to intervene in this process and control which pollen actually gets to your plant with hand pollination, but that's really only for very small scale production and for breeding. It's not something you would normally do in a big way, even in your garden, unless you were really concerned, or you had some especially important seeds, or you had more than one kind of corn in your garden. Then you can use hand pollination and keep the varieties separated. But it would be too expensive to grow very much corn this way. Other than that, to prevent pollination that you don't want, as I said, you plant further away, you plant cornlet flowers at different times, you plant border rows that you don't harvest for seed, and in that way try to avoid contamination with anybody else's corn, and especially this transgenic type, as you know, those genes are not allowed and they're patented, and most of us don't want to have those unless we are producing that corn for sale, I guess. So there is another part to the story. So, you know, reducing unwanted pollination is one thing, but once the pollen lands, there is also a way to intervene. And it has been used successfully for a very long time in the world of popcorn production. If you've never wondered, popcorn is mostly grown in the corn belt. Popcorn is corn, and it would normally easily cross with dent corn and lose its popping ability right off the bat. So how the heck did they deal with that? Well, they use this interesting trait called gametophytic incompatibility, and they are able to grow popcorn in the corn belt even surrounded by all this transgenic field corn. So gametophytic incompatibility, it has a long story actually. Some genetic researchers noted in the 20s that some popcorn could be used to pollinate and set seed with some other popcorns, but if you reverse the process and took pollen from those other lines and made crosses, you wouldn't get much seed set. And they call this cross incompatibility. And they were quite fascinated and actually immediately set to figuring out where this trait was found and which chromosome it was and which other traits it was linked with on that chromosome, and worked a lot of that out well before DNA sequence was ever understood. By the 1950s, crossing incompatibility was found in many South American and other popcorns, and it was being bred into commercial popcorn varieties to prevent crossing with dent corn. So it looked like a pretty handy trait if you think about it. If yours you know, blows pollen wherever, who cares? But as long as the other guy's pollen doesn't get onto yours, then you're set. So commercial popcorn really took off, and popcorn quality increased by using this trait. By the 1970s, breeders had developed white field corn with cross incompatibility using the same trait from the popcorn. And breeders in the Soviet Union also used this trait during the 80s to protect high lysine varieties from outcrossing, which would reduce that quality. Lysine is an amino acid that would make the corn much more useful as a protein source. In the 90s, breeders found this trait in some tropical field corn varieties, and different genes for this trait were found in teosinte, a wild weedy relative of corn, which sort of looks like corn in a very small way, but mostly looks like a grass. And then in the early 21st century, breeders began using these genes for crossing compatibility in field corn to protect organic corn from crossing with transgenics. So I'm going to explain how this works and some of the genetics and the breeding behind it, because I have a feeling some of you may want to make use of this trait out there. Certainly I'm going to explain how you can. So there are now 3 known genes for this trait: Gametophytic Incompatibility 1, Gametophytic Incompatibility 2, and teosinte crossing barrier 1. Each of these genes is found in corn, and corn normally carries 2 copies of the recessive version of these genes. However, in popcorn, where we found GA1S, and in teosinte, where we found GA2 and TCB1, there are some dominant alleles that change how the corn plants interact with pollen from other corn. And there are alleles or variations of these genes that are very, very strong at preventing other corn from setting seed unless they also carry the exact same gene. Well, that's pretty handy. GA1S is the allele used for protecting popcorn, and it's found in quite a few popcorns out there. The other 2 have just recently been found and are being used to very small degrees in breeding work right now. And if you don't know David Podol, he grows Dakota black popcorn, which does seem to be a GA1S carrier. All right. Corn carrying these genes just looks like corn. This is an inbred with GA2S, and it just looks like an inbred, whatever. You can't tell it's there unless you see some effect taking place with the crossing. So in some respects, you have to take somebody's word and do some test crossing to determine whether or not the trait is actually there and whether it's homozygous, having 2 copies and being fixed, or if it's heterozygous and we're not carrying one of these strong alleles at all. So how does this work? It's really kind of fascinating, so bear with me because we're going to get a little sciencey here. Here are some graphs showing the percentage of pollen tubes that could grow to certain distances down the silks. So we've got silk distances at the bottom, starting from right there to 2 centimeters, 4, 6, 8 centimeters, and then getting down near the egg sacs, and then percentages on the left side. If we look where the number 1 is in the upper right corner of that graph, what we see are pollen tubes coming from plants that carry GA1S pollen will readily grow tubes all the way down to the ovules on plants that are homozygous for GA1S. So the GA1S plant easily works with the pollen carrying this gene, recognizes it as pollen, helps that pollen tube grow, and fertilization takes place. If we look at number 2, we can see that pollen with the normal recessive gene for GA1 struggles to grow. It doesn't grow very long tubes. Very, very few of them get very far, and once we get close to the egg sacs, we find just a very small percentage of those pollen tubes were able to grow. A few of them grew more than 3 inches down the silks, and that essentially for this plant showed that the GA1S was very protective to keep unwanted GA1 pollen from setting seed, whereas it readily allowed GA1S pollen to set seed. Okay, so how else does this play out? If we have a plant that's heterozygous for GA1S and it just has the 1 copy, we get a similar pattern where GA1S pollen will readily grow tubes down to the ovules, and we still see some reduction in the ability of the recessive GA1 pollen to grow. It will grow, but it's at a competitive disadvantage even if this plant carries only 1 copy of the GA1S. And then if the plant is homozygous GA1, it's a normal corn plant like we'd see in the Corn Belt, both types of pollen easily grow tubes right down to the ovules and will set seed. So normal corn can be crossed by any of these, but corn that is homozygous for GA1S really only wants to be setting seed when pollinated by GA1S pollen. This is a drawing from that paper just to show on the right is normal pollen tube growth. So we have a pollen grain landing on a little silk hair, and then down our little diagrammatic silk, we'll get that pollen tube to grow and it'll go right down to the egg sac and we'll get fertilization. On the left, we have all kinds of problems. The pollen's been landing on here. Growth stops early. Growth gets misdirected. Sometimes they'll grow down and then change direction. They're just completely lost. This is what happens when GA pollen, the recessive type, lands on a plant that's carrying GA1S. So that sort of pollen is at a competitive disadvantage for actually setting seed with the plant that is carrying GA1S. So as I mentioned, back in the 70s, they had released some white field corn lines with GA1S, and they found outcrossing with yellow corn grown immediately adjacent to their plants was reduced to a fairly low level, from nearly none to 1.5% on average, although I recall one cross had as much as 7% crossing with the yellow corn. It was way, way less than the 80 to 100% you'd normally expect to see. So this is a really effective tool for reducing unwanted outcrossing, even with corn that is immediately adjacent to the corn you're trying to grow. And as all of us should know, that's not what we ought to do if we want to grow pure seed. If we want to grow pure seed, we want to have some distance and border rows and other things to reduce the crossing already. But this tool can be added to that and make it even that much better at reducing unwanted outcrossing. This table comes from a paper in 1955 by a geneticist named Thomas, and he simply laid out what it was all the popcorn breeders were learning about what would happen if you crossed plants of different genotypes as far as seed set and the genotypes of the progeny. Normally we expect normal Mendelian genetics to take place, and those are fairly predictive of what kind of genotypes we would see in the progeny. But when GA1S is present, we don't get normal Mendelian genetics. It really messes it all up. So what I'm going to do is walk through this somewhat diagrammatically rather than just going through this table. So in all cases, we get pollination. Pollination is just when pollen lands on the silks. But will we get fertilization and seed set if we use pollen from a normal homozygous GA plant and put it onto a homozygous GA1S plant? It has 2 copies of GA1S, as you can see. So the pollen moves from tassel to ear, and we don't get seed set. Awesome. That was what really threw them when it started, is they saw this, and that's not normal for corn. Corn is usually very, very willing to accept pollen from anywhere. So what if we reverse that? Homozygous GA1S plant shedding pollen and going onto a normal homozygous GA plant. We get normal seed set, and all of the offspring will be carriers of GA1S. That's pretty standard Mendelian genetics there. What about normal pollen onto a plant that is heterozygous for GA1S? Well, as I showed you in the earlier graph, pollen tubes don't grow as well, but they do grow, and corn sheds millions and millions of pollen grains, so a few will get through and only a very few are necessary. So yes, we will get normal seed set. Half of the seeds will be carrying GA1S, and half of them will be homozygous normal, which again follows pretty normal Mendelian predictions. If we put pollen from a carrier of GA1S onto a plant that is homozygous for this trait, so we know it's resistant to small GA pollen growing, we get seed set, but only from the GA1S pollen. So we have now screened that out, and we're not getting normal, the normal pattern of inheritance here. Only the GA1S pollen is setting seeds. So it's kind of a screened it right out. If we put pollen from a heterozygous carrier onto another heterozygous carrier, one that has only one copy of the GA1S, we also get seed set, but we also don't get normal Mendelian genetics. We should expect to see a quarter of the offspring be homozygous for the dominant gene, a quarter of them to be homozygous for the recessive trait, and then half of them to be carriers. That's the normal genetic segregation we see when making such a cross. But in this case, because of the way GA1S works, we're going to get 1/3 of the offspring be homozygous for GA1S and 2/3 be carriers for the gene. So, since we're all geniuses, we immediately see how we could use this to our advantage, no doubt. But luckily, there were some geniuses who took a look at this data back in the 30s and 40s, and by the 50s had figured out what to do with it in order to breed varieties that are homozygous for this trait that you cannot see, which is a pretty tricky deal, but still regularly used by popcorn breeders and others right now. So, if we're going to breed this, we want to breed this trait into something we like. So whatever our local variety is that isn't carrying this, if it's an old popcorn that doesn't carry it yet, if it's a sweet corn that we especially like, if it's a flint corn or some other kind of field corn that we really like, if it's some inbred lines we like because we're going to make some hybrids to sell, whatever it might be, we're going to take some local corn that is homozygous normal, small GA, and cross it with pollen from a GA1S source. And the result, as I already showed earlier, of this particular kind of cross is offspring that are all carriers for this GA1S. Well, one cross means that the seeds are half the genetics of our local variety and half the genetics of whatever the source was. That's probably not where we want to end up. So we'll probably do some kind of back crossing for a few years in order to recover the genotype of the corn we like locally, but with this new gene. This is a classic breeding program. And so we're going to take those crossed seeds and cross them onto our local variety one more time. We'll get seeds that are 75% our local variety, but unfortunately, because this new cross now is only from a carrier onto a normal, half of the seeds that result will be carriers and half of them will be homozygous normal. Well, that's a problem because you can't look at them and tell which is which. Well, once someone sat down and started thinking about this and looked at that table that we have, they saw, aha, I see how to do this. I can easily do some test crosses and figure out who's got it. So they did something that corn breeders do sometimes, which is called paired full sib crosses. Okay, so before I go further on that one, a full sib is when we have one father and one mother. Now that's sort of how it works each time mammals reproduce, or most mammals. But corn, because it is an ear with all of these many silks can have a family on that ear that has many fathers and the one mother. But if we're going to carry pollen and do hand pollination, we can take pollen from only one male and put it onto that ear shoot, and now we know that all of those resulting kernels are full sibs. They have just one mother and one father. Okay, and these are going to be paired because we're going to do this. We're going to take pollen from a number of these plants from this mixed population now that are carriers and not carriers. For each plant, we're going to take pollen over onto a homozygous tester, write that on the bag so we know which plant it came from. And we're also going to go to our local variety, which is normal, and put pollen onto that and write down which plant it came from. Then we'll go to another plant and another and another until we have made paired full sib crosses with enough plants from this mixed population to be sure that we've probably found at least one carrier in the mess. Carriers are 50% of them, but it doesn't mean you're going to find them all right off. If you've ever flipped a coin, you know that sometimes you can have quite a string of heads all in a row before you finally get a tails. So we'll do enough of these paired full sib crosses, so hopefully we'll found one at least that is a carrier and have moved along this cross the way we want. The result will be some ears that have half carriers and half normals, and other ears that are all normals. We'll also have some new homozygous testers that are 1/4 of our local type, which may or may not be of interest if you really like generating new tester varieties. At any rate, once we have done this, we get stuff like this. So here are some paired full sib crosses. At the top, we have the homozygous tester that had the GA1S, and at the bottom we have ears from the normal local variety. And in this case, we don't see the parent, the male parent, because it was just used for males. But in each case, these are the pairs. So plant number 6 went in both directions. I think it's plant 10 in the middle, and then plant 13 on the end. And there were 0 kernels on that ear, and they didn't bother to send that. They just sent back a little tag that said no set. What it shows is that the plants that were normal had very few seeds set on the homozygous GA1S testers, or no seed set. So we know these those male donors didn't have the gene, so we won't save any of the seeds in this case. However, there were some others that did set seed well on the homozygous testers. So plant 1, plant 4, and plant 5 all set seed on the tester, and we can save seeds then from the normal plants at the bottom, and we know that those are half carriers and half non-carriers. So right back where we were, but we know we've got carriers, and we can continue our process. So this really does work. It's pretty neat to see. I'm excited because I've been working on this for years, and I'd really like to see it working. So we're going to continue exactly what I showed you, making these paired full sibs until we've made 6 generations or more of crosses and have recovered 63/64 of our desired genotype or more in the pedigree of these seeds. And we know some of these seeds are GA1S carriers. So we found our variety. We've got carriers. What do we do next? Because we have to finish this up and get them to be homozygous so they breed true, and we've got this new variety that acts like the old one but is homozygous for GA1S, so we can protect it easily. Well, we're going to switch gears a little bit and do 2 more generations. We're going to do half of those paired full sibs. We're going to take pollen from the mixed carrier population, cross it onto the tester, and then also self those plants, which is something plants can do. They're hermaphrodites, both male and female, so a corn plant can actually set seed with itself. That doesn't work in mammals. It's not how our biology is. And what we'll find here are some of these plants set seed on a tester and some didn't. The ones that did do have seeds on their ear that are, because of the self of a carrier onto itself, we'll get 1/3 of those seeds are homozygous and 2/3 are carriers. Anything else that we have is just normal, and we're not interested in that. So how do we find out which of these are the homozygous ones? Because you can't tell by looking at it, and now we can't use this tester trick because part of those seeds are carriers and part of them are homozygous. So they will all set seed on a tester next time. So this time we go back to our table, or at least our geniuses did, and they came up with a neat new trick to help finish up the process. They took corn that had blue seeds and was normal, homozygous small GA, and they used it this way. We know that homozygous plants are very strongly resistant to GA recessive pollen. So what we'll do is we'll take this pollen from the normal blue corn, put it onto these ears that are of this mixed population one day, and the following day, after giving them a real good head start to grow pollen tubes, we'll self them one more time. The result will be some ears will be most all or completely yellow, and we know those were homozygous GA1S. Those are the ones we'll save. The other ears that were only carriers with a full day head start will have enough blue corn tubes grow through that will have these blotchy yellow and blue ears, and we'll chuck those out. We'll save the homozygous GA1S plants, and the process is done. This is the kind of stuff breeders do sometimes, and takes a little while, but it's very beneficial for humanity, so makes you feel good at the end of the day. So my question to all of you, not that you're going to take this and run at this very moment. I'm sure you would want to ponder this and perhaps contact me for copies of what I just showed you if you want to do it. But, once you understand some of these techniques, what magnificent thing could you breed yourself and bring to the world? What I just showed you are things you can do in your own garden, things that people have done in their own garden for thousands of years. You can breed your own corn and make something special happen.
So Frank,
Yes.
Are there other opportunities for like the folks that are online or the folks that'll be listening to this in the future, which we hope will be tons, to help you with this or any other sort of breeding programs that you might be connected with?
Of course, and I will be explaining a little bit about that here in just a moment.
Oh, good. Just didn't want you to forget that opportunity to advertise.
Yeah, well, thanks for the plug, Sue. This photograph in front of us is of some of the variation of maize from Peru. And as you can see, they have some magnificent corn, and we can breed stuff like that too. It doesn't have to be blue and purple and red and spotted and all those colors, but it could. Still taste yummy. Okay, there's one downside to using this gene, because every tool has to be managed. So here is the downside. If our neighbor's corn, which is transgenic and normal as far as GA is concerned, is pollinated by our GA1S corn, which it could be if the wind blew that way, and it produced volunteers in their field, those volunteers would be GA1S carriers and potentially transgene carriers at the same time. And as we know, if it's GA1S, it can pollinate our corn the next year, and it could carrier transgenes into our corn if that occurred. So these volunteers that are possible in our neighbors' fields are a potential threat. And the story for corn, of course, always is rotate. Rotate your fields, encourage your neighbors to rotate, encourage everyone to get rid of volunteers, and don't let this happen, and this tool will be exceedingly useful for a very, very long time. Okay, so how are people actually using this? Well, the popcorn breeders have been doing it for a very long time. Most popcorn varieties you can get have GA1S. They're at least carrying, if not homozygous, for the trait. Some commercial breeders are breeding field corn with GA1S. This includes the Pure Maize brand corn that you may have seen. With OFRF, Organic Farming Research Foundation support, I am breeding GA1S and GA2S into a range of maturity as field corn for other breeders and farmers. And there are some public breeders and farmers out there who are starting to play with this in their field corn. And then as far as sweet corn, I don't know of anyone doing this with sweet corn yet. It's possible it's been used out there, I just haven't heard of it, but certainly could be used. So you could grow sweet corn that didn't outcross with other corn that didn't carry the gene. And you could join in any of this at any of these steps as well. So increasing things to get them out to people once they have the traits, or helping them make crosses, those things are opportunities that exist. And I am releasing seeds with these traits that will hopefully be useful in our region for people to do just this. So whether it was a collaborative process with myself or something you wanted to start on your own, these things are here. I know where the seeds are to be found. I know the techniques. Always happy to share how to do this, because this is a useful thing. Now, it is important to remember, however, that if you're growing popcorn and sweet corn and field corn and you cross GA1S into all of them, then you aren't going to get outcrossed by your neighbor's corn, but you would probably outcross among all of your varieties, because now they would all carry the same thing. So one has to sort of put this trait out there in a strategic fashion to help meet whatever your needs are without sort of undercutting your future ability to grow things separately. However, if you had GA1S popcorn already and bred GA2S into your sweet corn, you could then at some point, I believe, mostly grow sweet corn and popcorn with little distance between each other and almost no likelihood of outcrossing, which would be really a fun improvement from where we are right now. So just to quickly wrap up what I have to say about this, organic and traditional corns, these are valuable crops. These are really wonderful things. We need to protect them. Corn is a wind-pollinated crop. It will always be so. Transgenic corn, therefore, is an ongoing threat to the economic value of organic corn and our ability to maintain traditional varieties of every sort. Gametophytic incompatibility, the inability of these pollen tubes to grow on corn carrying some of these traits, has been used for decades to prevent unwanted outcrossing in popcorn. And this trait is being bred, and we can all take part into that breeding. Breed it in any other corn we like. It just takes some time. So I'd like to acknowledge my OFRF funders, the USDA National Plant Germplasm System and the Maize Stock Center. Both provided me with seeds that I have used. A number of corn breeders who've taught me a lot of things along the way. Walter Goldstein, Major Goodman, Jerry Kermichael, who's a maize geneticist, identified the new traits coming out of teosinte, then Margaret Smith, corn breeder in New York, and Ken Ziegler, a popcorn breeder from Iowa, and many other researchers that were listed in a handout that I can send to Sue if you'd like to share a handout around. So, great big dziękuję. Thank you to all of you for listening to me drone on. Now, if you've got any specific questions, I'd be so happy to entertain them.
Yes, you send me that document. I certainly will put that out when I get the YouTube video up. And Ariana wants to know where she can find this person's popcorn seed so that she can start growing some of her own.
Oh, popcorn seed that carries GA1S?
Yes.
Several popcorns carry that already, and as far as I understand, I believe the Dakota Black popcorn that David Podol is selling from Prairie Road Organic Seed, I believe that carries this gene, may be fixed for it. Steve Zwinger is selling Pinky popcorn. I think that's sold out of Irish Eyes, and he is pretty confident that Pinky also carries this gene. There are also some popcorns from Iowa that carry it, but Pinky and Dakota Black are two varieties you could probably grow in our region without too much trouble. Where is Ariana located?
She lives in Surrey near Minot.
Okay, that's pretty far north. Well, Pinky would probably be the one to try first because I think it's a little bit earlier. And then, I don't know, maybe we could poke around sometime and see what else is available. It wouldn't be too hard to figure out which other popcorns carry the gene because all you'd have to do is find one you can grow and then make some pollinations onto a known carrier at some point and test it out. And that's something that I could certainly help you get started if you wanted to figure it out.
Feel free to unmute yourselves and ask questions now. Frank will be with us for a few more minutes here. Travis and Ariana, you have my permission to speak if that's what you're waiting for.
I was thinking if I just grew the popcorn, and I don't know that I'm going to get into the crossing of it, but if I could just get the GA1S seed and be able to grow it that year, and then, like you said, maybe take a year off of it or something and grow it again in another year. I tried growing popcorn this year just from a catalog seed, and it really didn't work.
No, almost all of the popcorn available is commercially bred is for the central corn belt, and it's just too slow. Beautiful plants, but as I'm sure you saw, they start to flower in mid to late August, and it's just way too late.
Well, it seemed like it grew, but it wasn't. It was more like field corn.
Well, it's tall.
No, but I mean, it had heads and everything. It just didn't. About one of them looked like popcorn. One ear looked like popcorn. The rest looked like sweet corn.
Oh my! They did have see, whatever popcorn they were selling obviously doesn't carry this gene, and it got badly outcrossed. That's too bad. I would highly recommend trying out Pinky and see what you think. Certainly, if you do, let me know. Steve Swinger thinks it probably has this gene, but I could probably test it out some point over next winter if it worked for you this summer. How's that sound?
That sounds cool.
I don't actually grow corn here in the winter, but I do send it to nurseries down in South America over the winter, and we could do some test crosses and see if it worked. Figure out what we had.
Yeah.
Any other questions or ideas?
Right.
Just asking if there were other questions or ideas. Happy to entertain you while you're here.
So seriously, where did you get that picture of that guy sawing off that corn husk?
Well, corn used to be really big back in the day.
Right. Oh my gosh.
Before Photoshop and before computers, people played in the dark room and did some amazing tricks. And this was, you can see there's some funky lines on there. I photocopied this out of a Seed Savers Exchange book, probably about 15 years ago, maybe more. And they had been putting these out from old postcards that people sold around Iowa back in the early 1900s, and this one guy just made lots and lots of these. This was by far the absolute best. It's perfection. The man actually seems to be standing on a leaf, and his saw really seems to be cutting into that ear of corn.
Definitely. Gives me an idea, though. I love Photoshop, and I love cards, and we're always working on ideas for the Tri-County Tourism Alliance, and I got have collected a lot of old black and white farm photos from the Germans from Russia, the interviews that I do. And
sure.
Yeah, I'm like, God, we always wanted to do a series of cards with funny sayings on to make money for the group, but it would be funnier to do things like this.
Sure. Wouldn't it be fun to have a group of kids climbing up a stalk of wheat with sickles?
Oh, God. Or, yeah, anything. We got all that old weird farm equipment, and Yeah, it would be great. You know, the best pictures of vegetables I ever found were from the research station south of Mandan. Man, they got some good old trial variety pictures, cabbages and things like that. There's always a copyright though with that kind of thing. So,
Well, this one, I'm sure whoever did it is long since gone, so off it goes. But there other photos in the series included, people with wagons full of a potato, and an army of people cutting into a melon the size of a house, and all kinds of crazy stuff. So yeah, people have always liked to have fun. And
fun with vegetables. We should do a curriculum. Fun with vegetables. I kind of like that idea. Of course, it's grant writing time, and my head is filled with ideas. But hearing no more questions, I'd like to thank you all and really thank Frank for that absolutely way over my head presentation on corn. But that's okay. I just love growing vegetables. I don't feel the need to know it any deeper than my love relationship with them. So we're glad to have people like Frank on our sides. And if you do want to do seed breeding or vegetable trials or any other kinds of things like that that we can help you out with, don't hesitate to contact NPSAS or farms, and we'll get you hooked up with the right people. So with that, any more questions? Nothing? Comments? I will bid you adieu and enjoy that nice weather out there. I've been tempted to go out and plant some lettuce in the hoop house.
Thanks so much. The sun's just broke clear again. It's just going to be an amazing May day again.
Yay! See you guys.
Bye now.